Hi all, Dr. Clark here again. Today, for a general biology lecture, we're going to talk about organelles of the cell. Okay? And so organelles, if you remember, are just membrane-bound compartments. And when we talk about eukaryotes, these membrane-bound compartments have different kinds of functions. So in some cases, they might be um, areas where carbohydrates are broken down, like the mitochondria, or proteins and lipids are broken down there also, nucleic acids. Okay? They might be areas where carbohydrates are constructed, like chloroplasts. And they might be areas where digestive enzymes are housed, okay, to help with the breaking down of either foreign objects coming in or food particles through phagocytosis. Okay? And so, at any rate, we're going to talk about different organelles, the importance of the organelle to the system, and ways at which you can kind of visualize them or see them under a microscope, if that be the case. Okay, so here you can see a cell, and you can see the nucleus of the cell is right here, and you can see these small organelles in different regions, okay, and they're occurring along these protein fibers, which are causing them to what we call cytoplasmic stream, so they're moving throughout the cell. And we'll look at this again in much greater detail as we progress through the course. Okay, so first I just want to talk about a couple things. So the nucleus, okay, is, probably the most well-recognized organelle in eukaryotes. Remember, prokaryotes do not have organelles, and they don't have a nucleus. Okay? That doesn't mean they don't have DNA. Prokaryotes do have DNA. Okay? They just don't have membrane-bound DNA sources. Okay? And so membrane-bound, what we're talking about is, for the nucleus, is something we call the nuclear envelope. Okay? So the nucleus, where the DNA is housed in the nucleolus, the region where ribosomes are made, is wrapped around a double membrane, okay, called the nuclear envelope. So what's the function of the nucleus? Okay, most people know this. It's the place where information is stored. Okay? What kind of information? That's the information that is utilized to construct proteins. Okay? So I want to make sure that people understand the purpose of your DNA, and really the purpose of any organism's DNA. It's to store the blueprint to make the proteins that build your body. Okay? DNA is not making proteins. Okay? It's just the instruction manual on how those proteins should be put together. Okay? And when we progress through the course, we'll start talking about situations where you might need a new enzyme, or you might need more transmembrane proteins, or you might need more muscle proteins, and all kinds of situations where you might need one of the 150,000 different proteins, okay, that can be coded for by your genome. Okay? We'll talk about how that is then constructed. How is it put together? Okay? But back to the nucleus. Okay? So when we look at the nucleus, this is the site where your DNA is stored. The other pieces of the nucleus, like the nucleolus, is a distinct region where ribosomes are made. Okay? And so this is very important, okay, from the standpoint of protein synthesis. Okay? So again, the purpose of your DNA is to be able to synthesize proteins, okay? And then the purpose of your nucleus is to hold that DNA along with other components that are utilized in the construction of proteins, ribosomes, RNA, these kind of things. Okay. So here you can see the nucleus. Okay, the nuclear envelope is that membrane that wraps around it. The nucleolus region. Okay, this is where ribosomal RNA will be constructed and portions of the ribosome will be built. Okay, here it's showing you that you can start to see some chromosomes kind of packed together. Now remember, most of you probably have seen the pictures of chromosomes where they kind of look like X's. Okay. Now, only during special times in the cell do chromosomes actually condense enough for you to be able to see them. Most of the time, most of the time your DNA is loosely packed. Okay, it's not condensed down. It's long strands. Okay, and you can't see them even with a microscope. It's just not possible to see the DNA. Okay, it's at times where this nuclear envelope is going to go away. It's going to dissolve, and your chromosomes are going to condense, and then your cell is going to split, divide. Okay. At those times, your DNA, your chromosomes will become condensed, and you can visualize them. Okay, so during periods of mitosis and meiosis, and we'll come back. We're going to study mitosis and meiosis, in a little bit later on in the lecture series. Okay, so the next structure that we need to talk about is in direct connection with the nuclear envelope. Okay, and so normally it is, wrapping around the nucleus of the cell, and that would be called the endoplasmic reticulum. There are two types of endoplasmic reticulum. We'll go over both. Okay, the first one I want to go over is called the rough ER. The next one is what we call the smooth ER. Okay, the rough ER gets its name because it is loaded with ribosomes. And that should tell you right away what the purpose of the rough ER is. Okay? And so that is the site of protein synthesis. Okay, so if we go back to ribosomes, ribosomes are the site where messenger RNA comes in, lays a blueprint down to link amino acids together. The rough ER has ribosomes attached to it. Okay? And it is going to be this membrane. It's going to be this membrane that can vessel out, can take little vesicles and move them to another entity. Okay? Golgi apparatus. Typically it's the Golgi apparatus because your stuff that's made in your cells need to be stamped with a sugar coating that indicates that it was made inside and it's self-made. Otherwise, your immune system will attack it. And we'll come back to this. Okay. So rough ER, again, is associated with the nuclear envelope. It's the site of ribosomes, okay, and protein synthesis, and is really distinguished by its look because it looks like kind of fingerling projections that come out. I'll show you a picture of it with little dots all over it. So here you can see that the rough ER, and you can see in this picture here the smooth ER, and we'll get to that next. But the rough ER has, these ribosomes associated with it, so proteins are going to be made. So all you have to do is take and copy your DNA into messenger RNA, and then it moves into the rough endoplasmic reticulum and starts making proteins. And these proteins get packaged into these little teeny vesicles. So here you can see some vesicles that are moving away from the rough ER. They're going to be moving into the Golgi apparatus for repackaging and sorting and things like that. Okay. So the smooth endoplasmic reticulum, because it lacks ribosomes, you know right away that it has nothing to do with protein synthesis. Instead, okay, this is a area where things like fatty acids can be synthesized, phospholipids can be synthesized. It also supports the storage of different types of ions and things that might be utilized in the production of phospholipids and fatty acids. And in some organisms, It also can be associated with breaking down damaged lipids or poisonous lipids, depending on the organism that we're talking about. Okay, so the smooth ER is very different than the rough ER. It's not just different because it has no association with ribosomes. It's also not directly attached to the nuclear envelope. So it's sitting out here off of the rough ER. So here you can see those vesicles. Again, same kind of system where this smooth endoplasmic reticulum is going to be constructing things like fatty acids and phospholipids. They're going to pass them in vesicles. These vesicles will move towards wherever the lipid is needed. Often that's membrane, so maybe a new organelle is being constructed, or maybe the cell's growing and you need new phospholipids to build the plasma membrane, or to add to the plasma membrane. These vesicles will be traveling to those regions and dumping off the new phospholipids that are being made. So here, that brings us to what we've already kind of talked about, and that's the Golgi apparatus, another structure highly important to pretty much all eukaryotes, and very important because this is the spot where everything that is constructed inside the cell gets labeled. Yeah, so a lot of people think of this as kind of like the UPS store. Material comes in, it sometimes needs repackaged. Right? So sometimes things like proteins need to be folded in this region. Other times you need to just stick a sugar coating on it, something like that. And then it gets put into vesicles and it moves out to wherever that protein, in some cases wherever that fat is needed. It might package a bunch of enzymes together and then send it out. There's lots of situations. Okay? And so part of this, and we'll talk about this because we already kind of talked about what keeps cells small and how you can kind of deviate from small cells, is by increasing the amount of surface area. Well, one way you can increase the amount of surface area is by getting a bunch of stacks or a bunch of folds of membranes. Okay? Golgi apparatus does this also in the form of cisternae. And so here you can see the function, like we already talked about, is to sort, ship, repackage, put sugar coatings on, make it so your immune system doesn't attack self-made products, and then ship that out through vesicles to whatever region of the cell that material is needed. So here you can see that exactly what I'm talking about. So here, maybe proteins were made in the rough ER, vesicles sent in, they go on the cis side of the Golgi apparatus, goes inside, they go into these cisternae folds, and in there they can get repackaged. So there is associated proteins in here that can do repackaging. There are other proteins that might put sugar coatings on them. There are lots of things that can go on inside the Golgi apparatus. When that material has been repackaged and is good to go, it goes out the trans side in a vesicle form, okay, and moves wherever it's needed. In some cases, that's all there is to it. Okay? That vesicle stays, and you got a bunch of proteins inside that vesicle. Things like lysosomes are made this way. So you get a bunch of digestive enzymes that are made in the rough ER. They're passed through these little vesicles into the cis side. They get repackaged, sent out the trans side. They stay inside the vesicle, and now you have this lysosome, which is an organelle, which with a bunch of digestive enzymes in it that can now be utilized to digest food, foreign invaders, these kind of things. So it's like the immune system inside the cell. Ribosomes. Okay? This is kind of a tricky one. because ribosomes are not organelles. So they really don't belong in the organelles section. Now they act like organelles, right? But because they do not have membranes, right, they consist of two subunits, a large subunit and a small subunit. But because they do not have a encompassing membrane, they are not organelles. Okay, so they're not considered an organelle. Now they might have functions like organelles, and in this case they do, okay, because they are the site of protein synthesis. Okay. The other thing about ribosomes which make them very unique, and we kind of talked about this already, is they contain their own RNA. And because all organisms on the planet have ribosomes, we've been able to utilize, researchers, scientists have been able to utilize this ribosomal RNA to connect the dots. What group is related to what group? And this is what started this ribosomal RNA research, is what allowed us to come up with the three domains, archaea, bacteria, and eukarya. Prior to that, when I was in undergrad and when I was taking general biology, there's no such thing as domains. Right? And so it's this new research in ribosomal RNA and whatnot that has allowed us to get a clearer picture of how things are connected. Ribosomes can be attached to the rough ER, and we already showed that. Okay? But they can also be free-floating out in the cytoplasm, or what sometimes is called the cytosol. Okay? And so you got this ribosome association with messenger RNA. The messenger RNA doesn't go away. We'll come back to this. Messenger RNA can be sitting in the cytoplasm. The ribosomes are sitting in the cytoplasm, and you get these feedback loops. If you need an enzyme or if you need a certain protein, that messenger RNA is triggered to find a ribosome, construct proteins. If there's too many of that, it often will have a negative feedback, and that messenger RNA gets bound up and it's not making any more proteins. And we'll come back to positive, negative feedback loops, biochemical pathways, these kind of things as we progress. So really the function of ribosomes, we've already talked about them, is protein synthesis. Okay? And so again, just to make sure it's clear, DNA and RNA are not making proteins. They're the blueprints. Okay? They're the instruction manual for how the proteins should be linked together. What amino acid sequence should be laid down. DNA and RNA are not making amino acids. Okay? It's just telling what amino acids should be laid down. Okay? And we'll come back to that. So here you can see a picture of the ribosomes. Again, they can be on the rough ER associated with that. They can be sitting out here in the cytosol inside the cell. And there's ribosomes that are associated with mitochondria that are self-made ribosomes, which help build proteins, essential proteins for things like the electron transport system and situations like that. We'll come back to that when we get to cellular respiration. But here you can see large subunit, small subunit ribosomes. Kind of looks like popcorn. And sitting out there in the solution or attached to the rough ER. Lysosomes, I already kind of mentioned them. They're found in animal cells and are really a kind of multifaceted system. So on one hand, they're great for digesting food. On the other hand, they're great for digesting foreign bodies. And so they're kind of like the immune system, but also the digestive system. And so if something gets captured inside the cell, lysosomes will attack it and dissolve it through the digestive enzymes that they put into the foreign body. If the cell itself engulfs something through phagocytosis, the lysosomes will come in, digest it, utilize the energy from whatever it was that was consumed. So they are, the main function is for digestion and waste processing, but like I said before, it's also sort of the immune function of individual cells. If you have a mitochondria that is old and the DNA might be degraded, lysosomes will digest it. But if you get, a bacteria that gets in to your cell, sometimes the lysosome will find that bacteria and dissolve it, or you'll have other systems which will find that bacteria and encapsulate them, and then the lysosomes will be called to digest that bacteria. So here you can see it, lysosomes. Really, they're nothing more than a membrane, okay, with digestive enzymes in it. And we're going to look at a little video in a second showing you lysosomes in action. But there are two processes by which lysosomes work. They can do what we call autophagy, which means that they're dissolving things from within. Okay, things that are already in the cell, maybe a damaged mitochondria, maybe a damaged Golgi apparatus, something, and they're going to go dissolve it and recycle those things. Remember, even though society, in most societies I would say, we're not very good at recycling things, our body is excellent at recycling things. We utilize metabolic water, we utilize carbohydrates, we utilize fats and proteins and nucleic acids over and over again. We utilize enzymes and proteins and things like that constantly over again. And if they're degrading, if they're being destroyed because of too much work, they're dissolved, and then the pieces of those are utilized to build new ones, to construct new ones. Our bodies are really good at self-maintaining, rebuilding material from internal compartments, internal storage, things like that. So you got this autophagy. That's one way that lysosomes can be called to action and dissolve things from within. And then you get things like phagocytosis. There are other types of cytosis like penocytosis, which we'll come to later. But phagocytosis, engulfing of solid particles, and then this is called a phagosome, and then that phagosome can be dissolved through lysosome action. Lots of organisms eat this way. So this is how they actually consume food. When we talk about things like protista, lots and lots of protista, this is how they eat. They engulf things, bring it in as a phagosome, lysosomes call to action, dissolve the material, okay? Or maybe not lysosomes, but something else which has digestive enzymes. Same general process though to dissolve that material, because not everything has lysosomes per se, okay? But they definitely will have digestive enzymes. Even plants have digestive enzymes. They don't have lysosomes, but they have another spot where they hold digestive enzymes and can have the same general effect.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. The hydrolytic enzymes degrade proteins, nucleic acids, lipids, and carbohydrates, and are formed in the endoplasmic reticulum. These enzymes are then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles, such as old mitochondria. This results in the destruction and recycling of these structures.
So again, really two ways. Autophagy, where things are already inside that need to be broken up, okay? And then phagocytosis, or some type of cytosis where things are brought in, food, foreign bodies, these kind of things can be dissolved and and the nutrients are utilized. Now, in some cases, maybe this is a poisonous substance and the nutrients don't want to be, the cell doesn't want that material in. Then it can just push that vesicle to the outer membrane, and it does that kind of reverse phagocytosis where it spits out whatever the material was, so it's not within inside the cell. We'll look at some examples of this later on. All right, so vacuoles, this is where I was getting at when I was saying, animals have lysosomes, but other organisms, have digestive enzymes and the same kind of properties. They can do autophagy, and in some cases they can do phagocytosis also. And one example of that are plants and fungi. They have vacuoles in which contains digestive enzymes. Now these vacuoles, depending on the organism, can have multiple functionalities. In some cases, it will be used just for like digestion of foreign bodies or food and things like that. You can think of things that have a fairly good vacuole system, digestive enzyme system, pitcher plants, Venus flytraps, these things. They're going to be digesting things that are consumed. Okay, so they have a lot of digestive enzymes that are just dumped in and dissolved, and then they can get nitrogen and phosphorus from the living organisms that are being consumed. Well, the dead organisms eventually that are being consumed. There's other situations, though, where that vacuole is not utilized for digestion as much as it is for storage. This could be storage of water. So when you look at vacuoles inside of like cactus and succulent plants, their vacuoles are full of water. And it's a very important purpose of the vacuole is to store water. In other cases, though, maybe you're looking at aquatic plants that water is not the issue, but nutrients or ions or different elements are the issue. So they might be storing calcium and sodium and some of the other essential elements or molecules that's needed for photosynthesis and cellular respiration inside their vacuoles to maintain those, because they're an aquatic system, and that's going to be leaving the system fairly fast. Water is not the limited resource, something else is limited. So it really just depends on the plant or the fungi and what habitat they live in, how the vacuole is utilized. And that's the cool thing about the vacuole, is it can be utilized in many different ways. So here you can see this vacuole. It looks really, when you look at them under a microscope, a lot of times it'll look like just a big air bubble inside the cell. It can take up quite a bit of space, but again, this is a membrane-bound organelle, and those membranes are still phospholipids. They have the ability to shrink down and expand, and that's one of the great properties when it comes to retention of water and things like that. That water can fill up the vacuole, it can push all the organelles to the outer edge, and then as the day goes by in things like desert plants, vacuole gets smaller and smaller, and it releases that pressure on those organelles, and it returns back to shape. And this can happen over and over again. That's the great thing about flexible membranes. All right, bringing us to mitochondria, something that most of you have probably heard of. Okay? Most of you know mitochondria as the powerhouse of the cell. So again, back to surface area and volume. Again, when we need to have lots of connection points, organelles and organisms and cells, they've all evolved to increase surface area if that's what's needed. And in one case, like the mitochondria, it's definitely needed. So in order to increase surface area, we can get these cristae, these sac-like regions inside the mitochondria that make up this mitochondrial matrix. And along these cristae, along these membranes, you're going to have a bunch of embedded proteins. We'll come back to this because we'll talk a lot about the electron transport system. But this is going to be the region of ATP synthesis. So when we talk about mitochondria being the powerhouse of the cell, it's the powerhouse of the cell because carbohydrates, proteins, nucleic acids, lipids get into the mitochondria, are transported into the mitochondria in different forms, and then they are broken down in different processes. We'll get to this. And that breaking down and free electrons is utilized in the electron transport system to drive the production of ATP. ATP is really just adenosine, so adenine triphosphate. So an adenine molecule, just like in DNA, adenine molecule with 3 phosphate groups hanging off of it. Now, that is important because that is the currency, that is the energy for your cells. And so this is where that's constructed. Mitochondria, very interestingly, has its own DNA. Okay? And that DNA is separate from nuclear DNA. And we call that mitochondrial DNA, or sometimes it's abbreviated mtDNA. And this is one of the neatest things that humans have the ability to do after the discovery of mitochondrial DNA is we can start tracking the female line. Okay? So if you think about this, mitochondria are inherited from your mother. So every single organism on the planet that has a mitochondria gets it from their mother. Okay? So whether we're talking about plants, whether we're talking about fungi, whether we're talking about animals, whether we're talking about certain types of protista, they're getting it from the mother line. Okay? And so that is passed down from mother to offspring, male or female, it doesn't really matter. Then what we've been able to do as researchers is, knowing that, we can track the female line back. We can see what did the mitochondria of really the first female Homo sapien look like. How many mutations has there been in maybe that 200,000 year period? Okay? How many mutations have we seen over 200,000 years in female mitochondria? And it's awesome, and it tracks us all back to Africa, where the original Homo sapiens originated. That's also where the fossils of the original Homo sapiens are found. And we'll get back to human evolution much later in the lecture series. But nonetheless, mitochondria has its own DNA. It's a site where ATP is produced. Right? And again, they have these cristae, these finger-like projections that make up the matrix of the mitochondria. Along these projections are going to be embedded proteins for the electron transport chain. Let's look at another one that has its own DNA, chloroplast. Okay? So just like mitochondria, double membranes, also got membranes inside that are stacked together to increase surface area, which we call these membranes or these fluid-filled sacs. We call them thylakoid membranes, and if you got a bunch of them, we call it grana, and then the liquid in between we call it stroma. But regardless, we'll come back to this when we talk about photosynthesis. These organelles have their own DNA, and it's inherited from the mother organism. So in the case of plants and algae, they're getting chloroplast from their mom, from the mother organism, and this is how they convert carbon dioxide and water into carbohydrates. So it's really the site of photosynthesis. We'll come back. We'll talk a lot about photosynthesis as we progress. So here you can see thylakoid membranes. Each one of these is a thylakoid membrane. And you put them together, you get a granum, a stack of thylakoid membranes. In between, you're going to have the stroma. This will play an important role when we start talking about photosynthesis. There's interesting pieces of the photosynthetic cycle that happen in the stroma, the space in between thylakoid membranes. Now, that really brings me to the topic of organelles that contain their own DNA. We've already talked about endosymbiosis, or this engulfing of things and not destroying them. So phagocytizing something, not destroying it, but utilizing it, forming a symbiotic relationship. And that's what scientists have pretty good evidence for mitochondria and chloroplasts and why they contain their own DNA. Why is it in circular form, much like a bacteria? So it's just a circular chromosome, and that DNA is not found anywhere else in any of the cells. Okay? So there's a lot of interesting pieces. The other thing is it must be pretty prehistoric. This must have happened a long time ago because the symbiotic relationship is extremely strong. We do not have the capability as of right now to remove a mitochondria and grow it on its own, or a chloroplast. Okay? And that's much like when we were talking about the symbiosis between termites and beneficial bacteria in their gut. Neither one can live without the other. It's the same thing in the organelle system. Neither one can live without the other. That doesn't mean that they couldn't at one point in time, that they couldn't, 1.5 billion years ago, that they weren't free living. Okay? But over a long period of time of this evolutionary connection between the two systems, now they can't occur apart from each other. All right, with that, hey, we're going to move into one more piece of cells, and that is cell structure. So how is it that cells don't collapse on themselves? What allows for cells to move? How do you get influx of material into the cell and out of the cell? That's the next topic that we're going to talk about. Until next time, later.